Hello to everybody. It is really a pleasure to give this talk, and I'm grateful to the organizers of this GEOTOP series for the kind invitation, to Carlos, DeWitt, and Renzo. And what I will tell you about is some work that has been going on in our group for a few years, where we try to use the concepts at the interface of mathematics, mostly physics, and biology, and apply them to biomolecules mostly. So I understand that this may be slightly different from the background that most of you have, so don't hesitate to interrupt me if you need some clarifications in what I will tell about. So probably a good way to introduce and motivate the problem that I will present is by recalling some experiments that have already been mentioned in a previous talk given by Professor Sumners in the series, where we heard about the spontaneous knotting of DNA that occurs inside the bacteriophages. So these are some slides from some striking work that has been done in the group of Joaquim Roca in Barcelona. Here in the top left, you see a picture of a bacteriophage. That is a virus that infects bacteria. And in this bacteriophage, most of the DNA is actually concentrated and packed inside the head of the virus that I'm pointing at with the mouse, except for one end of the DNA filament, which is anchored at the foot of the virus, ready to be injected into the bacterium. And it so happens that the DNA of this bacteriophage is double-stranded, but the two ends are single-stranded and complementary. They are so-called sticky ends. So when the two ends have the chance to go inside the capsids in mutated forms of the virus, which lack the anchoring point at the foot of the virus. So when the two ends go inside, what happens is that they anneal, and essentially you trap in the form of proper topological knots any type of physical entanglement that you had in the DNA molecule to begin with. At this stage, what experimentalists do is to blow away the capsid by chemical means. They collect all the genetic material and they run it through a gel using an electric field. And these molecules, these circularized DNA molecules that have all the same length, are found to travel with different velocities. These are the spots that you see on the right. They travel with different velocities, not because they have different length, but their migration velocity in the gel is different because they have different topologies. And people over the years have learned how to establish what type of knot you have depending on the specific velocity that you observe. And by this means, people have been able to establish that 95 or more of the DNA molecules inside the viral capsid are knotted, and also that there is an abundance of certain knot types, especially torus knots, compared to other knot families like twist knots. So that brings about several questions, because this is not only interesting per se to know that you have a lot of entanglement in the DNA inside these viral capsids, but it's very informative because this topological spectrum that you can measure, it's an indirect fingerprint of how DNA is organized inside. And so although you cannot still probe directly how DNA is packed inside, you can do that to some extent with cryo-EM images and so on. But this is a very convenient statistical way of finding what happens. And if you have different models for how the DNA is organized, then you can validate them by checking how well you reproduce the knot spectrum. And that is something that we did in collaboration with DeWitt some time ago. And so this is just to give you a flavor of what we did. You can consider different models for DNA, and then you can study also the ejection process and see what kind of agreement you have with the data. The point that I would like to dwell more into today is precisely what is the role of the presence of knots out there in the ejection process of the DNA from the virus, because as I mentioned, in the wild-type virus, all the DNA filament is packed inside the head except for one end that is anchored at the tail of the virus. And so if you imagine that inside the capsid you have a high probability of being entangled, a high probability of having a physical knot. As I told you, there is a more than 95% probability to have these open physical knots. What happens when the DNA gets out from the very narrow pore, so the narrow channel that you see in the micrograph? And the pore has essentially to come to terms, the knot has to come to terms with the narrow pore through which it cannot pass. You would imagine that the translocation process gets halted by the presence of knots. But this is not the case. These viruses are infectious, which means the DNA can get out despite the presence of the knot. And so looking at how tight knots translocate through a pore, it's an interesting question per se, and I will try to give you a perspective on that using coarse-grained models and simulations. Another motivation for looking at the problem comes from an ongoing effort that the community of biochemists have pursuing for the past few years, where people try to use solid-state nanopores in order to sequence single-stranded DNA. So instead of the double-stranded DNA, which is very stiff, that is inside the viral capsid, here scientists deal with single-stranded DNA, so only one part of the double helix. It's a much more flexible chain. And the idea that goes back to something that Dimer proposed at the end of the 80s is that you can use an electric field to drive these single-stranded molecules that float in solution, and because they are charged, you can use an electric field to drive them through a narrow pore, and if the pore is narrow enough, you can measure some physical-chemical properties that will be sensitive to the specific chemical identity of the nucleotides that are going through A, T, G, C. And in this way, people, for these observables, they use the ionic current, so they measure what is the flux, the ionic flux that goes through. If the pore is narrow enough, that will really depend on the chemical identity of the bases that are going through. And so people have had some success at sequencing single-stranded DNA. So remember, these are very long and flexible filaments. And people dream of sequencing uninterrupted stretches made of hundreds of thousands of bases, of nucleobases. And so when we heard of this effort several years ago, we thought that was a kind of dangerous thing to do, because if you aim for translocating through these narrow pores some filaments that are so long that it will be inevitably knotted, by the time that you drag them and pull them through this very narrow pore, which is only 1 to 2 nanometer wide, so it's wide enough to let just a single filament translocate and not more. So an entire knot could not go through because you would need at least 3 filaments to be dragged to the pore. Then we thought, you see, maybe people might actually consider not to sequence stretches longer than a certain contour length, because if you sequence longer ones, then you will systematically incur inevitably into knots, and then you will clog up the pore and you stall the translocation process. So we wanted to have a more quantitative understanding of the process, and we decided to use some coarse-grained molecular dynamics simulations to look at that. And I can give you a flavor for what these simulations look like. So what you see here in the left part of the image is a configuration, an equilibrated configuration of a chain of beads, fully flexible, that has been generated with Monte Carlo means. So that is a configuration that is equilibrated in bulk. It consists of 15,000 beads. And then we took these configurations, equilibrated by the computer, and we primed one of the ends close to this spot here, where there is a pore. Actually, the pore, you cannot see it because it's narrow in the image, but actually there is a little pore there, and where there is an electric field that drives the charged beads of the chain through the pore. So let me run the simulations and then the movie of the simulation, and then I will run it again and comment it again. So this is what the translocation process looks like. I hope it is visible in the online presentation. So let me run it again. And so what you see, now I'm stopping it at a certain stage. Let me repeat again. There is an electric field that acts like a motor that drags the DNA through the pore. This electric field is acting locally only here in the region that I'm pointing with the mouse, where there is the pore. There is no electric field in the bulk in the rest of space. And because because this model flexible strand is being pulled from one specific point, you see that the translocation process actually occurs through the propagation of a tension front that invades progressively and rectifies the chains, and then the chain is translocated and packed essentially below the chamber. So the reason why you see that the chain is denser at the bottom of the slab, past the pore, rather than at the top, is not because there is a bottom that the chain cannot go through, but it's actually because this translocation process occurs very much out of equilibrium, and the chain that has just translocated to the trans side has not had the time to relax, and therefore it looks much more compact. So there are all sorts of interesting physics that people over the years have elucidated for this type of system, and in particular one that I found perplexing the first time that I saw it is that the time that you need to translocate a chain at constant force that is applied at the pore grows with a power greater than 1 with the chain length, meaning that if you have a chain that is twice as long, it will take much more than twice the time to translocate it. And this intriguing effect is explained, the explanation is sketched in the right-hand side. I will not go through them with the scaling argument that is shown there. But the reason for that is that the polymer is embedded in space with a fractal dimension nu, which is the metric exponent, and it actually takes time for the tension to propagate through this fractally organized structure, and this is the reason why translocation time does not grow linearly with chain. So people over the years, I would say these are classic problems, so maybe even over decades, have studied the problem, and if you look at simulations that you can collect systematically on longer and longer chains, somehow you can really see that the theoretically expected translocation law as a function of the force that you apply in chain length obeys the theoretical relationship. There are still arguments about various regimes, various crossover, but I would say that the phenomenon is well understood. But I would specify is understood for chains that are free of knots. The theory, of course, cannot take entanglement effects into account because these are long-range effects that cannot be accounted in scaling theory. And so what our understanding is mostly for chains that are unknotted. What happens if you take a chain that has a physical knot and try to translocate it through the pore? So in our ensemble of configurations that we sampled in equilibrium with Monte Carlo simulations, we had some statistically knotted chains. And one instance is shown here. The green region is the region where you have a knot accommodated there. And if you tighten this region, you find out that this knot is the familiar figure of 8 knot. So what happens if I take this configuration and now I pull it at one end through the pore by the same procedure that I've shown you before? We want to remember that our goal was to characterize what maximum length we could address before running into the problems of having the pore clogged by a stuck knot. So this is a knotted configuration, this one here. I will start the simulation, show you how it behaves when you translocate it through a pore. And what you see is that there is actually no qualitative difference from what I've shown you before. When I saw this result, I was actually very surprised. Actually, when the result came on the computer, I actually looked at the raw data. I didn't watch the simulation, but I understood that the molecule had entirely translocated through the pore. So I said, something bad must have happened during the simulations. Probably we were pulling so tightly on the knotted configuration that we actually had a strand passage inadvertently happen in the configuration. But no, that's not the case. And I can show that by having a simulation run again, but with a close-up for what happens near the pore. And now, which you see here on the right-hand side, I will start the simulation again. So the knot is initially very high above the top of the slide. And after translocation starts, now you see the knot has tightened, has reached the pore. I hope it is seen in this web seminar that the quality, it's okay to see the effect. Let me show it again. After you start the translocation, the knot gets dragged and tightens at the pore entrance. It stays there as a sort of physical entanglement, but the chain is able to go through the pore anyhow because it's still capable of sliding along its knotted contour. So we were not expecting this effect at all, but once that you see it, of course, then you explain it to yourself, you accept it, and then you can also rationalize it. Essentially, the way that we explained it to ourselves a posteriori is that the thermal fluctuations that are always there were enough to keep the chain breathing by a sufficient amount that although the chain was pulled, like in a ratchet, thermal fluctuations were occasionally opening up the knot, and you were able to gain a little bit of an inch by inch of the knotted rope sliding along its knotted contour. However, when you understand this, you also understand that if you increase the force compared to how much thermal fluctuations can open the knot for you, then you might eventually prevent the knot from translocating at all, because essentially it is like increasing the self-friction of the chain. And this is what I'm showing in this slide here, where you see the same type of simulation setup that you saw in the previous slide, but this time the force is increased by nearly tenfold. And you see that now the molecules is not able to translocate anymore because the knot has become trapped at the pore entrance. The knot is there, you pull, but now there is no breathing room anymore that creates the slug that would be necessary to translocate through the pore. So all the phenomenology that I told you so far, which is for flexible, thin and fully flexible and long chains, it can be summarized by this graph that you see, where essentially you have on the x-axis, you have the translocation time since the beginning of translocation, and on the y-axis, you have the translocated fraction of the chain. If you start at low force, is the red curve, you see that the entire chain goes through with a certain amount of time. This is a knotted chain. If you increase the force, 10 piconewton is the green curve, you go through faster. But if you keep increasing the force, unlike what happens for a knotted chain, instead of going faster and faster and faster, you actually hit a plateau and you completely jump the translocation process because the chain is stuck due to the excessive tightening of the knot. And this is an effect that after we had seen it, has also been reported in other models for other type of biomolecules, not only polyelectrolytes, let's say single-stranded DNA, but also proteins and so on, and these are the references at the bottom of the slide. So probably this, as Carlo was mentioning, this would be a right time, 20 minutes, to maybe stop and ask questions about this first part of the talk, if there are any. Any clarifications needed? Okay, so I assume I can move on to the next part. So what I told you so far is for the behavior of a fully flexible chain, spontaneously knotted, dragged through a very narrow pore. A very nice set of results have been obtained experimentally by the group of Kees Dekker in the Netherlands, where they used, instead of a fully flexible molecule like a single-stranded DNA, they use a stiff molecule, double-stranded DNA, so the full double helical DNA, and they dragged it through a pore that was much wider, so 20 nanometer wide, and the pore is, the double-stranded DNA has a diameter of 2.5 nanometers, so this time the pore is 10 times wider than the thickness of the double-stranded DNA, so the DNA is thicker, is more rigid, but still they consider filaments of DNA that were sufficiently long to be spontaneously knotted, either physical knots in linear DNA or proper knots in DNA chains that have, after they are circularized in bulk. And they used a very ingenious setup to detect and measure the occurrence of knots. So the setup is actually illustrated in this picture here at the top. So the idea is that as the DNA filaments go through the pore, again, they are pulled through the wide pore by an electric field. You measure how much current is going through the pore. So you should imagine that you have a baseline current where the pore is not obstructed. So this current is brought by the ions that are in solution, and that they can go through the unobstructed pore. And so you measure a certain current. If you fish a DNA filament from the solution and you drag it through the pore, then of course there will be less surface through which the ions in solution can go through. And what you will see is you drop your measured current by a notch, because you have a filament that is obstructing the pore, and after the filament has fully translocated, then you go back to the baseline current. And so you have traces like the one that I'm pointing at with the mouse that are very easy to interpret. Essentially, you see a constant drop for a certain amount of time, and that is the duration of the translocation process of the single filament. And then you have traces that are more interesting to interpret, which are traces like this, where you see an initial drop by 2 notches, and then you go back to a single notch decrease, and then you go back to the baseline. How do you interpret this? Well, they rightly interpret it as the pore being entered by a backfolded DNA end. So the minute that the translocation begins, essentially you have twice the occupancy of the pore due to the backfolded end, and you have twice the drop in ionic current, and then you have only one after the backfolded end has gone through, because you have only one filament, and then you have 0 filaments, and you go back to the baseline. And then you have much more interesting traces, like the one shown here, where you begin like the process that you see here. The pore is actually engaged by a single filament, and then you suddenly see the current dropping by 2 units, 2 notches. Then you go back to a single filament occupation, and then back to the baseline. So how do you interpret that? An event that occurs in the middle of the translocation process. Well, the only way that they could make sense of what was happening was that this brief drop in current was due to the passage of a knot through the pore. And this technique you can use not only for physical knots in open chains, but also for proper knots in circularized chains, as it is shown by the bottom figures. And so this type of setup essentially was almost like a revolution in the field, because it gives access to quantities that you cannot really access in gel electrophoresis, which is very powerful, but can only answer the question of what type of knots you have, which is an important question per se, and what is the overall knotting probability. But with this type of technique, you can answer much more punctual questions for the type of process that you consider. So you can answer the question is, what is the knotting probability? But then other interesting questions that you can address are, where is located the knot on the chains that you translocate? Is it more in the middle, equiprobable throughout the chain, more probable towards the end? And also, what is the distribution of the lengths of the knots that you record? And these questions you can answer by addressing appropriate quantities that you can measure through the current. For example, by simply counting the incidence of these substantial current drop events, you have information about what is the incidence of knots. You just count in how many of the translocation processes you see these larger drops, and then you have a handle on the knotting probability. But then, not only you have that, but in this example here that I'm pointing with the mouse, you can ask yourself, so the knot passage has occurred at halfway of the process, at 75% of the process, in the top 25% of the process, and so on. So you have an information of where the knot is located on the chain. And you also have information about the size of the knot, because at least intuitively, you would imagine that knots that are bigger will take more time to translocate to the pore and will cause a longer drop in the current, a longer drop event. And they beautifully demonstrated the use of these experiments by pushing the characterization of the knotting probability to chain lengths of in excess of 100,000 base pair, which is a contour length that really you cannot address with gel electrophoresis, which I've always seen addressed for chains of the order of 10, 15 kilobases. So looking at the knotting properties of, let's say, lambda-phage DNA, which is a very sturdy virus, would really be beyond the reach of gel electrophoresis. But with this type of technique, you can address. And so we thought of using coarse-grain models in order to complement what had been seen in these experiments that I mentioned to you, and for which an explanation was not ready based on the available theoretical and experimental data. Because you record the current, but that's not like the same as visualizing what is happening. So you have an indirect information about the process. And so one interesting set of data that they collected was the distribution of the timing, distribution of the normalized timings at which the drop in the current, the knot passage event, was occurring. They saw that most of the knot passage events were taking place at a late stage. Again, this is something that is not really easy to interpret a priori. And then the other interesting feature they observed was that the translocation process, the duration of the translocation process, was biased towards very short durations. And again, that may be indicative that the knots are spontaneously tight, but you really wanted to check whether that is the only thing that is going on. And so we thought of using a model, actually a better model than the one that I've shown before. In the first part of the talk, when I was considering these fully flexible chains, I used a chain of beads. And then I told you that you could stall or slow down or not hinder at all the translocation process of a knotted structure, essentially based on how much friction you develop when you pull compared to the magnitude of thermal fluctuations. And so this notion that the internal friction of the molecule sliding along its set is important made us think that maybe we needed to have a model better than a chain of beads, because this is like the smoothest chain that you can imagine, and biomolecules, in fact, are all rugged, like barbed wire, essentially, and so they would have a higher hindrance of sliding along their own contour. And so we thought of using the most refined model that we could afford to use, given the length of the molecules that we wanted to address, which are thousands of base pairs. And so we settled for using this very nice model made by Doi and co-workers in Oxford and collaborators, which is called ssDNA. And it is a model that uses, for those of you that are interested, uses a 3 interaction center representation for each nucleotide. So there is a center for the phosphate, the base, and sugar groups, essentially. And it is rather detailed coarse-grain model because it allows the DNA strands to open if they want so. And so we thought to denature if they want to do so, to distort. And so we thought these were all relevant features, because the minute that we introduce a knot in a DNA strand and then we pull it and tighten it, maybe the DNA is going to behave in all sorts of bizarre ways due to topological constraints, geometrical constraints, and so on. So we wanted to have a model that is as detailed as possible. And so this is the counterpart of the simulations that I've shown you before, done for the models for double-stranded DNA. So the filament that you see here, it just looks like a one-dimensional, a very simple thread, but it's actually this double helical structure. And the red region, which I hope you can see, is where the knot is. So this is a circular model knotted DNA ring with a trefoil knot inside, so which we designed by hand. We let the chain equilibrate, and as in the previous simulations, then we primed the chain at the pore entrance. The pore is right here. I think it is visible. And then we started the translocation process by switching on an electric field inside the pore that acts like a motor that drives the chain through. So I'm going to run the simulation. Let me stop it. Okay, I will let it go through, and then I will play it again. So what you are looking at, now the chain has completed the translocation. So let me run it again and explain what you can monitor in the panels on the right. Essentially, the top panel tells you what is the fraction of the entire ring that has translocated as a function of time. So this reaches eventually 100% at the end of the translocation process. More interesting is what goes on in the bottom graph, where as a function of time, you see the location of the ends of the knotted region measured along the chain contour. And you see that that summarizes very aptly the fact that as the translocation progresses, then the knot tightens because these two curves approach each other. Then the knot is very tight. It makes a few diving attempts to dive into the pore. And when it has passed on the other side, it opens up again for entropic reasons. So like in experiments, we could, we had access to all sorts of measurable quantities that we could monitor in order to interpret also the experimental mimic and interpret the experimental data. And so, although we don't have explicit solvent, we don't have explicit ions, we can still measure what is the surface of the pore that is occupied by the strand that is passing through and by the pore. And essentially, you see that as a function of time, what you see, if you look at the red curve, that is the occupancy of the pore. And at the time when you have the translocation of the knot, you see a blip, which tells you that a lump is going through the pore, and you can measure when that passage event occurs, late, early, and you can also measure its duration, which you would expect to have something to do with the length of the knot. At least this is the intuitive expectation. And so just to remind you, this is the distribution of the timing of knot passage events that had been seen in experiments. And what we saw in our simulations is something that is quantitatively very similar. It's entirely analogous, I would say. We see a bias towards late passage of the knot. And the reason why you can explain it with a very simple model, a posteriori, of course, after you had the benefit of actually looking at what happens in 3 dimensions, which is a benefit that the experimentalists didn't have, of course. And the reason why you have this late passage event, in a nutshell, is due to the fact that after you initiate the translocation, due to the propagation of the tension front from the pore to the rest of the chain that I mentioned at the beginning, you have essentially a tightening of the knot that occurs starting from the end that is closest to the pore. So the knot is tightened essentially upstream. The knot is tightened and ends up where its most upstream end was at the beginning of the translocation process. So if you do these combinatorics with essentially, without even doing simulations, you just simulate this idealized process, you come up with a predicted curve for what the distribution of timing events should be, which accounts in a, let's say, satisfactory qualitative way for the fact that you should expect biases towards later stages of the knot translocation events. But then more intriguing is looking at what happens for the duration of the translocation events, which is something that you might imagine has to do with the size of the knot. And for that, we had big surprises. Well, first of all, if you just measure the duration of the events and you compute the probability distribution, that looks very much like the experimental one. You see biases towards short translocation events. However, if you look at the connection that these translocation events have with the size of the knot at the time that translocation occurs, then you see the data are really scattered all over the place. And it's really intriguing. You have, if you look closely, you can convince yourself that there are essentially 2 data sets that are superposed here. There is one set of data that has a very wide distribution of passage of duration, of the duration of the translocation process, and the associated length of the knot is small. And then there is another set of data points for which you have a almost linear relationship between the length of the knot and the timing, but the length of the knot can be very widely varying. So can we understand what is going on? Well, again, we had the benefit of having the possibility to look at the picture because we saw something that we had not expected. So meanwhile, you can try to guess yourself what explains the first set of data, the second set of data. Well, one set of data has really to do with the translocation of the knot in the way that you would intuitively imagine that happens. So you imagine that the knot, like in the simulation that I've shown, ends up on one of the strands that goes through the pore, it tightens, and then the reason why you have different translocation times, different duration events, has to do with the fact that if the passage occurs at the beginning or at the end, the velocity is different for the intriguing relationship between contour length, timing of the translocation, and velocity of the translocation. But then the second set of data, if you try to guess what happens, has to do with a different mode in which the knot presents itself at the translocation, and which you see here. And this, we call it a sort of delocalized mode of the knot for presenting itself. So if you ask yourself, where is the knotted region? I know that this is a sort of slippery concept from a topological point of view, but from a physical one, probably this is a question that our eyes are trained to see. If you ask yourself, where is the knot? That is, which is the shortest continuously uninterrupted the uninterrupted region of the chain, that once that you close it, you have the same topology that you started with, then you necessarily have to say that the knot is in the entire chain that is in the cis chamber, the one occupied before the translocation. And so if you look at this region here, you understand that what causes the occupation of the pore is not really the knot itself, but the depression in the current or the bigger occupation of the pore is actually due to a small region where the essential crossings of the knots are concentrated. And it is this region here, not the entire knotted region, but only the region with the essential crossings that gets detected when you do the experiments or do the simulations and measure the occupation of the pore. So essentially, the two sets of data have to do with the two different modes in which the knot can translocate through the pore, a tightened knot or a delocalized knot, but with a tightened region where all the essential crossings are conglomerated. Now, if you imagine to do these experiments, if you just imagine to close your eyes and drag through your fist the knot in the tightened version or in this delocalized version, you will, in both cases, you will just feel a lump going through your fist, essentially. And this is why you cannot really distinguish these two different modes of knot translocation when you do an experiment or when you do a simulation. So this really poses a challenge for future work, for trying to devise new type of setups that would be able to tell whether the knot is in the tight form on a single filament or delocalized and you just have a tightened cluster of the essential crossings in a knot that spans the entire chains. And I think it's, from a physical point of view, maybe also a biological one, it will really be relevant to distinguish between these two knot types, because as we heard in the talk by DeWitt Sumners a couple of sessions ago, the knots in DNA are actually targeted by topoisomerases that have the task to undo them. And whether you have a tightened knot or a delocalized knot, or whether you present all the crossings in a sort of compact region, really can impact the functioning of these enzymes, we believe. And also the elastic and mechanical properties of the chains are affected. As we all know, if you have a tight knot on a single filament, that weakens the mechanical resistance of the entire chain as opposed to a delocalized knot. So to summarize this second part, and I'm nearly done, I just have a couple of more slides to show you for a more recent simulations that we did on supercoiled and knotted DNA. But just to summarize this part, what I wanted to highlight is that theory, computation, coarse-grained models can really be a valuable counterpart to experiments in order to figure out what happens in the complex phenomenology of translocation of entangled and knotted molecules. Having the benefit of a simple model, if you can reproduce the qualitative feature that you see in experiments, gives you the possibility of using very simple physical criteria and insight in order to explain a lot of the features that are not directly accessible. And so for the very last part of the talk, it will just be a couple of minutes or a couple of slides. I would like to connect the translocation process to the problem that I mentioned, the simplifying action of topoisomerase enzymes on DNA. And because experiments by biologists have been performed on plasmids of DNA, so circular DNA of a few thousand base pairs, that can spontaneously in vivo, they can form knots that are complex. Pentafoil knots are not unusual. We wanted to understand what is the behavior, the physical behavior of these knotted molecules, and whether it was revealing of any features that might be actually exploited by topoisomerase enzymes in order to disentangle these structures. I just recall that one of the open problems on which much progress has been done, but probably there is still work to do, is how is it possible that enzymes that are small, that are a few tens of nanometer wide, which are topoisomerases, can have information about the topological state of long DNA filaments that can be micron long. And so how can you harness just some local information to disentangle the structure? If you just were to do local strand passages at random, it is known that you would end up actually knotting DNA molecule and not simplifying a knotted DNA molecule. So obviously the question is, what kind of physical clues, local ones, geometrical ones, structural ones, are exploited by these molecules in order to effectively simplify knotted DNA molecules? And so with this aim of contributing to understanding of the process, what we did in collaboration with Antonio Suma and Lucia Coronel was to study some model DNA rings that were supercoiled, as bacterial DNA is in vivo, and also knotted, because these knots can spontaneously be formed. And in particular, we consider pentafoil knots, 5-1, and also the twist knot of 5-2, so knots that are more complex than the trefoil. We looked at the conformational properties, but what I would like to tell you more about is some features that we had not expected to see, and probably maybe some clues, physical clues that can be exploited by topoisomerases, which is the following. As a function of time, we monitored our evolving supercoiled and knotted DNA rings and marked the regions that were knotted and also the regions that were supercoiled. So sometimes the supercoiling can just form a single superhelix, or you can have 2, you can have 3. And so as a function of time, we monitored, we colored in these chymographs where you have supercoiling, where you have the knotted regions. And in these 2 panels here, you have 2 different types of molecules. In the panel that looks greenish here, you see the trace as a function of time of the location along the chain. So 0 to 2000, that is a contour length coordinate, and you should assume a periodic boundary condition. As a function of time, we are coloring the position of the supercoiled region in, it's the knotted region, in a torsionally relaxed DNA molecule. So in the double helical DNA, we have put a nick, and all the torsional stress of the molecule could be relieved through this nick. So of course, the molecule gets rid of all the supercoiling that you put in at the beginning, and then you can monitor how the knotted region wanders along the chain as a function of time. And you can see that the knotted region is really mobile. In the plot that you see below, instead, we have an unknotted molecule, so we have no knot, but the molecule is not nicked, and so if you put in supercoiling at the beginning, then you introduce a constraint that the molecule has to deal with, and you can check and see where the supercoiling goes, how many superhelical regions you have and where they are, And you can see that in this region too, the colors change with a time over a time scale that is somehow comparable to the time scale over which the location of the knot changes in the previous plot. And then the most interesting plot is actually a plot where you have both supercoiling and knotting. So that is a molecule that is knotted in a pentafoil knot and also supercoiled at the same time. So for this molecule, you can simultaneously ask, where are the supercoiled regions and where is the knotted region? And you can already see by eye that the behavior, although the time scale is the same on all the graphs, the behavior is entirely different. Now, because of the presence of supercoiling, the knotted region is essentially locked throughout the entire duration of the simulation, which is the longest one that we could afford, and we estimate to be of the order of several milliseconds. And also the plectonemic regions are locked too. So it looks like having the interplay of supercoiling and knotting essentially locks all the essential crossings of the molecules and keep them stable, and presumably that is something that can really assist the action, simplifying action of topoisomerases, because it presents the crossings that you would like to target, so the boundaries of the knotted regions, and these are the crossings that presumably can be detected from their local feature, and especially they would remain stable as a function of time instead of changing continuously as you would have if you had in the graph below. So this concludes my presentation and my talk, and I would conclude by mentioning the people that at various stages have been involved in different parts of the work, in particular Antonio Suma and Lucia Coronel, that have been involved in the translocation studies, and it also includes the WIT for the work that we did on the spontaneous knotting of DNA inside viral capsids. And I thank you all for your attention, and I will be happy to take any questions.
Any questions?
Sure. Hi, Cristian.
Hi, Greg.
How are you?
I'm fine, thanks. And you?
Good. Excellent talk. Really nice. On the last part, you might be interested in a paper by myself and Lynn Zedrick, 94, I think, where we made an argument about topoisomerases using geometrical recognition of curvatures.
Yes.
And we also suggested in that paper that supercoiling would localize the places so that the topoisomerase could recognize the geometry.
Right.
So it's great to see that you have simulations.
Yes, yes.
So this effect. Really excited to see that.
Yes.
So it's very nice. That's just the point. So two questions. Here's a question. I don't remember the dates, but Andrew Belmonte, I think it was.
Yes, I'm familiar. Yeah, I'm familiar with the paper, with the concept of the hooked juxtapositions, and I think they. So just to intervene very briefly, I think that what we are adding here is some insight into the time scale over which these things can persist, which is something that is really important and that you need on top of the geometry, because if the geometry was only a labile thing from a temporal point of view, enzymes might not be able to recognize. But I'm a fan of the fact that the geometrical feature of these clasps are probably what really allows the molecules to detect and target the structure. So I'm completely on board with you on that, and I know the paper. Yeah.
Okay, great. So this is a completely different topic. On the pore thing. Andrew Belmonte did these experiments with these hanging chains that he jiggled.
Okay.
And what he found was that figure 8 knots would persist. You would see them, but the triploids would disappear really quickly. And The argument is something like that the figure eights require too much of the chain going back against itself to slide through. So I was wondering if you see that in your simulation.
Yes. So it's a very pertinent point. We have studied, it's a macro letters paper with Antonio Suma, I didn't show it. We have studied the effect of having different topologies in the tight pore translocation of the flexible filaments. And so essentially we were using a set of configurations that were all the same, and we were just changing the region near the pore, substituting one knot or another type. And what we saw is that torus knots, so we consider knot also composite knots. And we saw that torus knots are those that experience the least hindrance in going through. Essentially, they have only one choking point where the chain is highly squeezed on itself, and that is at the entrance of the pore, while twist knots, figure of eight, for the reason that you mentioned, actually ends up having two choking points, one at the beginning and one at the end. And that you also see in 5.2. And the translocation of these knots is completely different. So you jam them much more easily than the others. And so if I may add to that, another interesting feature that we really didn't know what to expect a priori, what we found when we looked at composite knots. So let's say that you have a 3-1 knot followed by figure of eight, or figure of eight followed by 3-1. And their translocation properties are different because somehow the tension on the chain decreases as you move away from the pore. And so the bottleneck is dictated by the behavior of the knot type that is closest to the pore. So having a trefold followed by a figure of eight has much less hindrance than the other way around. So it's commutative from this point of view. I hope I answered your question.
Yeah, that's wonderful. I mean, do you have this as a paper?
Yeah.
Have you published it? Can you hear me?
Yeah, so can you repeat?
Publish this work.
Yeah, it's published in macro letters.
Okay, I'll look for it.
I can send it. It's macro letters a couple of years ago, 2015 probably.
Oh, sounds great. Thank you.
Okay, thank you.
Any other question?
Well, just one question. Hi, Cristian. It is just in this translocation process. Is it possible that the knot is so big that it got clogged in your experiments?
In their experiment, I think in principle it could happen. The pore that they used, that Kees Decker used, is fairly wide. I think it's not exactly monodispersed, but it's between 10 and 20 nanometers. And so that would be enough to accommodate 4 or 5 strands, especially if they are tightly packed, in a cross section. So I think the chain length that they consider are not sufficient to get knots that are complex enough to clog pores of that size. But of course, I see no reason why they couldn't use smaller pores. But they actually wanted to avoid the chance of they wanted to have pores big enough to let the entire knot go through. But I think they estimated that most of the knots that they were detecting were trefoil knots, and I think the estimate is correct, and so those are too small to be trapped.
Is it possible to use this technique to detect knots? What type of knots, or for example, how complicated the knot is, just by changing the side of the pore?
I don't. I think that is an aspect on which pore translocation may not be that sensitive. I think in gel electrophoresis you can resolve very well through these bands, but if you use these very wide pores, I don't think that you would be able to tell a trefoil knot from a figure of eight knot, say. So I'm not positive that you have that type of resolution. You see other things. You see where the knot is along the chain and the duration of the event, but for the knot type, I'm not positive that you have a handle on that.
So, Cristian, a very nice talk. Did you do any simulations of duplex linear chains with knots in them to see if the knot would reptate off the end?
No, we haven't. No. I agree. It's nice. It's quite time-consuming to use these coarse-grain models, and so we really had to choose carefully the conditions that we were addressing, and couldn't really address systematically many ranges of forces, of translocation and so on. So we haven't. But I think, your question is very pertinent. For instance, a relevant question would be how many knots you lose through the ends, when they translocate, because they, in the same way that the knot travels in the ring but cannot get out of it, when you translocate it. If you do that in linear chains, then you lose them. And so these are chains that, although they are knotted, from their fingerprint on the ionic current, experimentally or computationally, you would say these are unknotted. But of course, you lose the knot, then you know that they had the knot at the beginning. So how many you lose? Your question would be very, we have not addressed, but I agree it's important.
Any other question?
So I was wondering, when you do this translocation and you get this signal, how sensitive is it to how many strands at the same time are in the material? Could you tell, like, at the moment there are 3 strands and now there are 4, or is this way beyond what you could measure?
I think in our simulations we can tell that, yes, because but in a way, in simulations you have some access to all kinds of structural observables that you have. So essentially we count what is the occupancy of a narrow slice of the pore, and you can definitely tell. So if whether you have 2 or 3 or more, yes, that you can tell. And in experiments, the signals are very clear. You see that these quantized drops, So in experiments, it's clear that you see them maybe even more clearly than in the simulations. For the currents, the ionic current, which we cannot monitor because we don't have ions, seems to be very sensitive.
Okay, so you can guess what knot type it is if you look at it. I think so.
But yes, because in the simulations, since we don't have the currents, we have to use some structural parameters, and in a way that is like cheating, because you have access to whatever resolution you want, to how much detail you want. So I wouldn't.
Thank you.
I would say we trivially can see that, yes, but I wouldn't attach any specific meaning, any deep meaning to that.
Okay. So, but in the experiments, you can see it too, or you can see it? No.
I think, so let me go back. In experiments, I think their quantized drops of the current were very, very precise. So,
okay.
Yeah. So you see here, for example, you see that you really go down by very precisely by 2 notches because you have 2 additional strands. So it's very precise.
Okay.
Even though it's very transient. So. My guess is that if I had sufficient computing power to simulate the solvent and the ions, by measuring the current, I would have the same sensitivity, and that would be very informative,
Okay. Thank you.
Any other question? Okay, then we really thanks for this wonderful talk, Professor Cristian Micheletti. I hope you